Can everyone hear me? Thanks for coming out today. My name is Randi Barton. I'm a master's student at Moss Landing Marine Labs in California, and I'm going to share a little bit of my master's research with you today. So I've been studying marine parasite dispersal capacity and how that relates to their transmission strategy, and I've been using population genetic analysis to better understand that relationship. And so most marine fishes and invertebrates have a bipartite life cycle. They start out with this larval phase that is highly dispersive, and they end in an adult phase that is more sedentary. And so that dispersive phase is really key in driving gene flow and their population dynamics as a whole. And dispersal is a crucial aspect for us in understanding how a species is going to persist given environmental change. And so while many studies have focused on the dispersal of fishes and invertebrates, few have done the same for the parasitic taxa, which is really interesting because parasites across the taxa have really diverse transmission strategies. Not only do they have larval stages, but they also utilize hosts to increase their dispersal. And most studies that are focusing on parasite dispersal focus so in the context of how it directly affects human health, but I'm also interested in how parasite dispersal affects ecology of important host species. In other words, we kind of need to know how parasites are moving in order to better understand what's happening to ecologically and economically important host species. Why do we care about parasite dispersal? Parasites are often overlooked when it comes to their contributions to community dynamics, but as we learn more, we're starting to understand that parasites are fully integrated within the system. This study looked at food webs in the Santa Barbara Channel Islands. They made one with and without parasites in them, so this one is just predator-prey interactions, and when they added parasites, the number of interactions nearly doubled. And parasitism is one of the most common biological interactions. It's actually almost every predator-prey interaction has some level of parasitism happening as well. And so because of this, we really need to understand what parasites are doing in order to understand the community as a whole. And because parasites have such a diverse array of transmission strategies, we can group them into two broad categories to kind of understand them better. There's the parasites that are direct transmitters. So these are parasites that use one host species to complete their life cycle. And there are the complex life cycle transmitters. Those are the parasites that use multiple host species to complete their life cycle. It also matters what's happening at a given stage. So they can have a narrow or broad host specificity, meaning how many hosts can they infect at a given stage. And the types of hosts also matter. So whether the host is sedentary or more vagile will affect their dispersal. So all of these variables can work together or separately to affect a parasite's dispersal. And an island system is a really great way to kind of look at this relationship. Because if you imagine you're a little parasite on a reef fish, your definitive host is your reef fish, which means that's where you spread your eggs. A reef fish is going to stick to the island that it kind of lives in. It's not going to travel hundreds of kilometers to a neighboring island. So you as a parasite, your eggs kind of stay maybe around that island. But if you're on a shark, that shark can swim circles around an island system. So you as a parasite get to disperse your eggs a lot further. And so island systems are a really great way for analyzing parasite dispersal because you can look at the connectivity of the parasites at each island, which will tell us something about how they're moving. And so my question is to look at the population structure of some marine trematodes across the northern line islands to see what that looks like. I like to think of the dispersal capacity of a parasite on kind of this axis of low to high dispersal capacity, where a parasite that utilizes direct transmission or has narrow host specificity or utilizes more sedentary hosts will likely have lower dispersal potential, will have higher population structure across the islands and low gene flow. On the other hand, a parasite that utilizes complex life cycle transmission, has a broader host specificity, or who utilizes more vagile hosts, they probably have a higher dispersal potential, which would be characterized by low population structure and high gene flow across the islands. So for today's focus, I'm going to focus on trematodes from family Microscaphidae, the microscaphodids. They are a complex life cycle trematode. So they move from a snail to a crustacean to a reef fish. So multiple hosts, more opportunities for dispersal. But all of these hosts are rather sedentary, so that kind of decreases their potential dispersal distance. So those two factors kind of work together to put them potentially medium to low dispersal potential. That's what I'm expecting. And so some expeditions occurred in 2019 and 2020 across the Northern Line Islands, also French Polynesia, but that's not included in today's talk. And Reef fish were caught at the fore reef of each island. Just for reference, these islands are located about 1,500 kilometers south of the Hawaiian Islands, smack dab in the middle of the Pacific Ocean. So these reef fish were brought back to the lab. In this case, these trematodes' definitive host is Acanthurus nigricans. It's a really common reef fish in the region. They were dissected layer by layer using microscopy to look for the parasites. They're very, very small, and they were sent to me for further genetic analysis. So for this, I stuck with mitochondrial CO1 DNA, and I just used Sanger sequencing. Because we don't know a lot, we're kind of starting out with the foundation of just CO1. And through all the trials and tribulations of parasite genetic work, if you know, you know, we ended up with about 88 trematodes across 3 islands, about 30 per island. And so first I ran some just general F statistics. And to jog your memory, an FST of 1 is when populations are completely separate. And so you can see we have a really high global FST of about 0.5. So that indicates that there's probably not that much gene flow happening. And I wanted to look at the haplotype data, which told me there's still some connection. So in this case, the white slices are unique haplotypes to that island, and the colored ones are the ones that are connected. And so there's a lot of unique haplotypes happening at each island. Kiridamati is the most diverse. Palmyra is the most isolated. I also made a haplotype network which showed that there's still very distinct clades, but there's still some connection happening. So we expected the dispersal capacity of these trematodes to be somewhere in the middle, and what we actually saw was much lower. That's likely tied to their host, as we thought. The hosts are very sedentary, they're not going to travel that far. And that little bit of gene flow that's happening could have happened in the past, could still maybe a little bit be happening through surface currents and eddies. So the Northern Line Islands are located right above the equatorial current. Kiridamati is kind of right at the top of the equatorial current. It sweeps up towards Palmyra. The red arrow points to Kiridamati, the most southern island. Surface currents and eddies can bring increased energy to this area. Studies have shown that there is larval connectivity due to surface currents in this region. This study simulated 20,000 particles moving through just by surface currents. And what they found was Palmyra, the most northern island, received about 3 to 11% of its particles from more southern islands, which would make sense with the direction of the equatorial current. And though this is simulated, it's also been seen in other studies about the survival of larvae through these big gaps in the ocean. So the Eastern Pacific barrier is a really deep part of the ocean. It's about 5,000 kilometers deep. Darwin referred to it as impassable for most marine species. And a lot of studies have come out showing the difference in populations across this barrier being completely different. But some studies have shown the opposite. They've shown populations of the same species to be genetically identical. For example, this study looked at my trematodes host, Acanthurus nigricans, and they were identical on either side of the Eastern Pacific barrier. They suggested that the time it would take for the water parcel to move across the gap is probably too long for that fish larvae to live, but times like El Nino and La Nina bring increased energy to that currents and can get them through. And so this has been tested in fish larvae, but not really in parasitic larvae. And parasite larvae, parasites in general, they usually release a ton of short-lived larvae, so they're probably not often surviving these really long treks across the ocean, but it's possible some are getting through, and just a really high mortality rate is decreasing gene flow. But we need to talk about the potential limitations here, right? Sample coverage. Are we just not seeing it because we didn't sample it enough? And this is a problem across parasitology in general. You can go out, collect a bunch of fish, bring them back to the lab, and you don't really know who's there until you get there. And funding doesn't always allow you to go back to get more, right? And along with the physical challenges of parasite sampling, there's also laboratory challenges. And if you want to commiserate after, I'd be glad to. We started out with 700 trematodes and ended up with this 88 usable for this analysis. So parasites have or parasitology in general has a fundamental lack of biodiversity knowledge, and that translates to a lack of protocols, reference genomes, so most parasite studies require an optimization period. And in that optimization period, you tend to lose parasites because they're so small, you have to use them, and then you lose them. So my main takeaways: life history can give us clues as to how a parasite is dispersing. but they have to be taken into the context of the physical factors around them, like the environmental factors such as surface currents and eddies in a marine context. And overall, parasitic taxa just need more attention in population and community ecology. For us to answer these questions, we need to have more foundational knowledge. It's kind of like a catch-22 we're in right now. And so my next steps, we are going to be conducting genome-wide sequencing. The CO1 was kind of just a foundational step. As I said, there was really nothing to go off of, so I spent a lot of my master's just figuring out how to get DNA out of them. But now we're collaborating with Hasselt University to do some genome-wide sampling, so talk to me in about a year if you want those results. I would like to acknowledge the Sandin Lab at Scripps, who did all the fish collections, the Wood Lab, who did all the fish dissections, headed by Katie Leslie, and all the undergrads who helped me through the RHIME program go through all that painstaking laboratory work of DNA extraction after DNA extraction. And I would also like to thank all my funding sources. Thank you. I think I have time for questions. Yes.
That was really interesting. Thank you.
Yeah.
The potential for dispersal between in the local area, Elmira and Kitty tomorrow.
Kiridmati, yeah.
Is there a potential that, as much like in Matt's talk of maybe the garbage trucks moving flies 20 kilometers, could a boat be moving from the southern island?
That's a really good point. So this study is an ancillary study of a more question like biodiversity related to fishing pressure. So they're looking at the differences in fishing pressure across the islands, removing hosts from the environment, how that affects the parasites. And so that's a really good point. Some of these islands are more active in terms of their fishing in general. I haven't looked at that yet, but that's definitely a good point. That's actually helpful, thank you. Yes?
Just to clarify, so when you're sampling them, you're sampling the parasites from within their host?
Yeah, within their host, yeah.
Do you know much about like the genetic specificity of that interaction, and are you able to distinguish between parasites not being able to disperse those long distances versus they are, but they're so locally adapted that they can't actually infect once they're there?
That's a good question. So that's another part of my thesis, actually, because cryptic species is a big problem, and because you can't tell the difference a lot of times between morphology. And what we found, because I looked at the Northern Line Islands in French Polynesia, there's actually larval samples of the French Polynesian species, but there was no adults that I found in the Northern Line Islands. So there's some interspecies competition, some barrier that's happening that doesn't allow the larval species to become an adult. So yeah, I'm not sure. Yeah. Okay. Thank you.
Okay, hello everyone. My name's Calum. I'm a graduate student in the Queller-Strassman Lab at Washington University in St. Louis. And today I'm going to be presenting some preliminary research that I've been doing over the last few months, basically talking about potential trade-off between genome reduction and interspecies competition in a group of facultative endosymbiotic bacteria. But I hope I'll first be forgiven for taking a step back to present my research in a wider context and reflecting that many organisms engage in symbioses with other species, with symbiosis simply being a term we're using to describe interactions between species that potentially have fitness consequences. These can be fleeting, consequential interactions based on happenstance within a generation, or they can be like fundamental aspects of either partner's biology. Symbioses, when they're maintained over many generations, can lead to long-term evolutionary consequences for their interacting partners. And this is especially pronounced for endosymbionts, which are species which live inside other species. These consequences can be morphological and phenotypic. Take, for example, the pair of wasps that you're seeing, where as a consequence of living inside a fig for its entire life cycle, the male doesn't look very much like a wasp anymore. They can also be genetic in consequence. What you're looking at now is the free-living organism with the smallest known genome, and compare that to a collection of endosymbiotic genomes, and you can see they're much smaller, right? Which makes sense because for these endosymbiotic species, they're living inside hosts. They have a much more simple, stable environment. They're free from the dangers that a free-living organism might be exposed to through predators, parasites, or fluctuation environments. So in our lab, we tend to look at evolutionary questions through the lens of our focal system, Dictyostelium discoideum. which is famous for its social life cycle, where upon starvation, a bunch of unicellular cells will aggregate together into a multicellular fruiting body, which then disperses to new environments, hopefully where there's food. And around 15 years ago, our lab discovered that there was a group of bacteria from the genus Paraboccodaria that effectively came along for this ride. They were able to infect cells in the unicellular stage and then maintain that association through this life cycle. We further discovered that there was 3 symbiotic species of Paraboccodaria that could do this, Bonia, Heliella, and Agricolaris, and that all 3 readily infect all the strains of Dictyostelium that we have tested so far. They're generally considered facultative in that we can culture them by themselves in the lab, and we can do the same for their hosts, but there are some slight asterisks there, and not for the Paraboccodaria, a lot of those aspects haven't been fully studied. So within our 3 species, they actually represent 2 independent transitions to the same symbiotic life cycle with Dicty, and do pretty much the same thing in terms of their effect on the host. With Heliella and Bonia looking like they're a much more derived evolutionary ancient relationship with Dicty, and Agricolaris kind of arriving to this relationship at a much later date in their evolution. And these phylogenetic lines along these phylogenetic lines, we also see variation in genome size. So Paraboccodaria agricolaris, I've given you a crude representation of its genome on the board. Around about 8.7 million base pairs long, it's pretty standard for a Paraboccodaria. We can compare that to Heliella and Bonia, which have a significantly reduced genome. Over 50% of their genes have been lost. Their genomes are around 4.1 million base pairs long. And this genome reduction has functional consequences. So if we look at the COG categories of these genes, so their function, we see that Agricolaris tends to cluster pretty much where you would expect with the rest of the free-living Paraboccodaria, but Bonia and Heliella represent true outliers in this group. And this genome reduction has especially had consequences for carbon metabolism. So when we tested a bunch of these strains of Agricolaris, Heliella, and Bonia, it was found that, while Agricolaris is pretty good at digesting most of various carbon sources, Bonia and Heliella are pretty goddamn bad, and they have lost the majority of their ability to metabolize most carbon. So my main question, based on this background, is whether Paraboccodaria with reduced genomes suffer increased fitness costs in interspecific competitive contexts with other bacterial species. And this is a question that's kind of rooted in explicitly biological aspects of this system, because it was found recently by former graduate student Trey Scott that when Dicty fruits, many Paraboccodaria are left behind in the stalk or in the surrounding substrate. And if there is a trade-off between this genome reduction and this competition, these left behind bacteria now, with their host having evacuated, are now vulnerable to competition with other bacteria. And my hypothesis is that would be the case, that we would see reduced fitness of the Paraboccodaria in competition, and their ability to actively reduce the fitness of their competitors would also be reduced. So how did I do this? So we selected 9 Paraboccodaria strains, 4 Agricolaris, 4 Heliella, and 1 Bonia. There's just less Bonia strains available. And these can be divided into the reduced and the non-reduced categories. With Agricolaris being the non-reduced and Heliella, Bonia being the reduced. These were then fluorescently tagged with a TN7 insertion vector to express RFP, which gave us the ability to discriminate between them and other bacteria in co-culture. We also had some competitors. So we had 6 species of soil bacteria that were isolated alongside Dicty. So these represent bacteria that these Paraboccodaria may well encounter in nature and may compete with. As we did with the Paraboccodaria, we fluorescently tagged these guys, but this time with GFP, which allows us to explicitly discriminate. We generated cultures in a 96 well plate and collected data in a microplate reader. And a basic competitive situation kind of looks like this. So we would generate a 50-50 mixture in yeast glucose media of an RFP and a GFP strain, and compete them for 48 hours at 24 degrees, which represents a kind of typical temperature that they might exist in the wild. And every hour with the microplate reader, we would collect optical density, GFP, and RFP fluorescence data. We also had some controls. So we first had an unlabeled control where we would compete each fluorescent clone against basically itself without the fluorescent tag, which would control for the particularly negative aspects of competition. And we had an alone control where we would simply just take that fluorescent bacteria by itself and basically give it the optimum environment with no competition with other strains and see how well it did. This is kind of an exemplary example of how one of these runs would look. So the red growth curve at the top is this alone, and then each of the colored lines below are growth curves under various competitive conditions. And basically, to extract fitness data from these are competitive curves. They vary a lot. It's hard to get stuff like just traditional doubling time measures. But to extract fitness data across this range of these curves, we can do something by calculating the area under the curve, which is basically the integral of the growth curve. And if we do that first for the alone condition, this is the maximum fitness that this bacteria can reach under these conditions, and then compare that against a competitive condition, we can divide the area under the curve in competition by the area under the curve when they're alone, which generates a relative growth measure for how that bacteria acts under competition. So do parabacteria with reduced genomes suffer increased fitness costs in competition? Here's some results. So on the x-axis here is the different competitor soil bacteria. So they're just abbreviated, so AT, BP, CK. And on the y-axis is the RFP, that normalized RFP ratio we've just talked about. So the higher it is, the better the parabacteria is doing. And for these three soil bacteria, it's Acrobacterium, a Brucella, and a Calum Strain, we see that they're pretty competitively benign. And the reduced genomes, the reduced genomes are in blue, no, the reduced genomes are in green, and the non-reduced genomes are in blue. And actually overlaid behind them is their wild-type isogenic competitors that we can compare their competition against. And what we can see is that none of these three soil bacteria are really hurting these parabacteria, but the non-reduced parabacteria are doing particularly well with these soil bacterial strains. But that pattern isn't consistent across all strains. If we then look at Pseudomonas and Serratia, we see that whilst the non-reduced symbionts are not doing particularly badly, the reduced genomes are actually suffering an decrease in their fitness. Finally, with Staphylococcus, which was the 1 gram negative bacteria, the gram positive bacteria that we used here, it just does. No one does well. It's pretty nasty. It decreases the fitness regardless of reduction status of the genome. Because of the nature of the experimental setup, we actually have the inverse of their perspective. We can look at how the soil bacteria are also doing with the different parabacteria, which is shown here. And I don't want to spend too much time on this. I mean, you basically have the GFP bacteria on the x-axis, and now we have the GFP competitor normalized growth on the y. But the one I want to specifically, and overlaid on each bar, is the wild type for that specific GFP bacteria, the wild type control. I specifically do want to focus on this Pseudomonas though, because if we pull both sides of this relationship together, on what would be your left-hand side, you have the normalized growth of the symbiont, where for parabacteria, the reduced symbionts are harmed, but the non-reduced symbionts seem unaffected. At the inverse is if the Pseudomonas on the right-hand side performs well with the reduced symbionts, but poorly with the non-reduced. And we can get further context on this through explicitly looking at the growth curves. So this is the Pseudomonas' perspective. This is how Pseudomonas is doing. In this example, this is how Pseudomonas is doing with the reduced symbionts, so the Heliella and the Bonia, against the alone condition, which is in this gray. And you can see that there's maybe a slight fitness decline. It's not growing quite as well as it does when it's alone. But for the most part, it seems its growth is kind of consistently across all. There's no explicit patterns that we can see that's hindering its growth. If we compare this Pseudomonas chart to how Pseudomonas does with the non-reduced symbionts, it's a much different pattern. So at around 10 hours, the Pseudomonas just starts to die off. And this also coincides, if we had the graph, with when the Parabocardea, the Agricularis populations actually start taking off in the culture. So it seems like whilst Heliella and Bonia have kind of lost their ability to kill off this Pseudomonas strain, Agricularis has retained it, likely as a result of its larger genome. But we can pull both these RFP and GFP results together if we explicitly divide the normalized growth of the Parabocardea, this RFP normalized against its alone, against the same value for the soil bacteria in a given well. We can look at the ratio of the RFP to GFP growth, or alternatively, just Parabocarderia's fitness compared to its soil bacterial competitor in the same well. So if your value is high, above 1, then the RFP bacteria is doing proportionally better than the GFP bacteria in that sample. And the inverse, if it's below 1, then the GFP bacteria is doing proportionally better than the RFP bacteria in that sample. We get this relatively convoluted graph. On the x-axis, I have divided the parabolicodaria into their reduced and non-reduced categories, and on the y-axis is just that measure, so that's the relative fitness of the parabolicodaria. So the normalized RFP-AUC over the GFP-AUC. And I have this one value where they're both, they would be doing equally as well in the dotted line. And you can see that for the most part, the non-reduced is doing pretty much better than the reduced. So the non-reduced, at least this is across all the GFP strains. They're colored by GFP strain. And we did a generalized linear mixed effects model to analyze this data and found that yes, the reduction status was a strong, had a significant and strong effect. The non-reduced ones did much better than the reduced ones. But this wasn't consistent across all the GFP strains. If we then disentangle this graph and look at individual GFP strains, we find that, especially for Staphylococcus, the one that we already talked about, pretty much always is suppressing the growth, and it's pretty much always doing badly, doing very well, regardless of reduction status of the parabolicodaria in competition with, which I'm reiterating here. So in summary, for D-Discardium-associated parabolicodaria, those with reduced genomes are poor interspecific competitors compared to those with no history of genome reduction, but this pattern did depend heavily on the GFP strain studied. With Staphylococcus, the Staphylococcus strain we used, there was no significant effect of reduction status on competitive ability, and the parabolicodaria always suffered. However, for the Pseudomonas strain that we had, there was a strong and significant interaction effect. It did very well with the reduced symbionts and very poorly with the non-reduced. So with that, I would like to thank my lab. I would especially like to thank Isra and Trinity for their general help in this process. And I would also like to thank the undergrads Emily, Elle, and Caitlin, who especially did the laborious task of trying to transform these strains when they had a really innate resistance to a lot of the antibiotics we were trying to use to select them for, and generally thank to my funding sources. So thank you.
All right, I'll just get started really quickly. So hi, I'm Youngseo. I'm a PhD student at U of T, and University of Toronto, and I'm going to be talking about my project, which is on investigating the mechanisms behind vaccine-driven virulence evolution of malaria parasites. So I don't think I need to do a lot to convince this room that malaria is an important disease. It has a huge human cost, and there has been some progress towards elimination, as you can see. There has been some progress towards elimination, but since 2015, we have been falling short of the milestones set by the WHO. And there are a couple of reasons for this. Well, there are a couple of challenges that we face when trying to control malaria. One of them is coverage, insecticides resistance, but another one is the evolution of resistance against drugs, so anti-malarial drugs. So on this figure here, the beginning of each red line shows when each drug was introduced, and the X's mark the first documented treatment failure due to resistance evolution. And it's pretty worrying, but you can see that resistant variants are found shortly after the introduction of a new drug, almost like every time. One good news is that malaria vaccines have launched. So the first malaria vaccine started rollout earlier this year in Cameroon. A lower cost vaccine started rollout in the Ivory Coast earlier in July. And there are many others in development. And thankfully, vaccines have stayed more robust to parasite evolution. So, stark difference from the slide I just showed, these lines extend to 2016 and the present day, but evolution of resistance does happen, as you can see in some of these X's. And this is because vaccines are not perfect. And what I mean by that is that, well, living through the past few years of the pandemic, we are now very aware that vaccines are not perfect. They're not 100%. So unless vaccines stop everyone from getting infected or transmitting to others, vaccine-driven parasite evolution is still possible. And this motivates my research. The inspiration for this project came from this paper. This is a study done on rodent malaria lab mouse model, which is often used because it shares some characteristics with human malaria. Here they did experimental evolution using a parasite antigen called AMA1, which is a malaria vaccine candidate. So in this experiment, they had two treatments, one where parasites evolved in vaccinated mice and control where they where the parasites evolved in sham vaccinated mice. And they passaged these parasites through either of these types of mouse for 21 passages or 147 parasite generations. And after that, they did like a common garden experiment where they put these evolved parasites in a non-vaccinated mouse and collected infection dynamics data. So one of the key findings from this paper is that vaccine-evolved parasites cause more severe anemia than the control. So in other words, they became more virulent. And this is a figure from the paper, and it shows minimum red blood cell density. So lower numbers here mean more severe anemia. And you can see that the vaccinated treatment caused more severe anemia. So, but interestingly, there is no evidence of sequence evolution at the vaccine target sites, the antigens. Because of that, we don't actually know what evolved and like what specific parasite processes evolved in this study. And my objective is to identify the differences in parasite traits and their interactions with the host that could explain the vaccine-driven evolution of increased virulence. And to do that, I take a mathematical model and then fit the model parameters to experimental data, and I do some comparison. I compare the parameters between the treatments to figure out what actually evolved. So before going into what I actually did, I'll first just briefly explain the parasite life cycle. It begins when free-floating parasite life stages called morozoites invade the host's red blood cells, like this. After invasion, these parasites replicate inside the red blood cell, and they become filled with multiple parasites. And after 24 hours, all infected cells burst to produce morozoites, like this, and the cycle repeats. So this process is this whole cycle takes 24 hours, and it is synchronous, which lets us use a discrete time model of red blood cells and infected red blood cells to model it. And of course, there are more processes happening in this model rather than just like the parasites replicating. So I'll just go over some of those processes. First, there's red blood cell turnover. So there is density-dependent and independent red blood cell production. There is also background death of these red blood cells and infected red blood cells. And we have two different types of immune clearance that model innate immune responses within the mouse. So there's indiscriminate immune clearance that kills both infected and uninfected red blood cells. And there's targeted immune clearance that only targets infected red blood cells. So there, I fitted 6 parameters. There's the, I fitted the 2 types of red blood cell production, the activation strength of the 2 types of immune clearance, background red blood cell mortality, and parasite burst size. So I fit these parameters to parasite red blood cell and parasite densities from days 3 to 16. And on the bottom here are some example mice data. So these are the red blood cell data, and these are the parasite data for the 2 treatments. And these are some example model fits, and they follow the data pretty well, I think. So this is what that looks like for all the mice. The black lines are the data, and each of the colored lines are model trajectories of parameter sampled from the posterior. And this is what that looks like for the parasite density. So now that I've convinced you that the model actually fitted to the data somewhat well visually. I also wanted to look at which parameters are actually different between the treatments. So each of these plots are parameters and they're colored by the treatment. And many parameters seem to have like some differences, but I want to draw your attention to these 2, which are very dramatically different. So this one here is background red blood cell production rate, and this one is the activation strength of indiscriminate immune clearance. So they're both lower for the vaccinated treatment than the sham treatment. So I did a PCA, and this is the biplot that I got out of it. And you can see that the sham treatment and the vaccinated treatment are kind of separated along this direction. And the axes, these 2 axes are the 2 parameters that I just highlighted. So in other words, compared to the sham treatment, the vaccinated treatment parasites are somehow activating the killing of infected and uninfected red blood cells less, and they're also somehow inhibiting the production of red blood cells. But how? How can we explain, interpret these findings? So firstly, what can explain the vaccine-evolved parasites escaping immunity? So first, there could be changes in other, So first, the antigens that are targeted in this study are expressed on all of these stages of the parasite. So they're on the merozoites and they get shuttled to the surface of the infected red blood cells. But there are other antigens that could have changed that we just don't know about. That we just don't know about, that either reduce immune signaling and detection, or immune signaling or recognition by the host, or the parasites could be producing less of other types of pathogen-associated molecular patterns, which can be recognized by the immune system. So secondly, what can explain the lower background red blood cell production of mice infected with vaccine-evolved parasites? So one might think of something like red blood cell production as more like a host trait, but we know that Plasmodium genus parasites can interfere with red blood cell production in the bone marrow. Another potential explanation is rosetting. So infected red blood cells the surfaces of the infected red blood cells can get sticky, and they can stick to uninfected red blood cells, forming these structures, rosettes, called rosettes. These are large, rigid structures that can get filtered out by the spleen. And if vaccinated treatment parasites produce fewer rosettes than the sham treatment parasites, there would be less splenic clearance and there less background red blood cell replenishment required in the vaccinated treatment. So, how could we test this experimentally? First, we could measure the propensity for rosette formation. So we would see more rosettes in the sham treatment compared to the vaccinated treatment. We could also measure red blood cell age structure. So since there is a slower turnover of red blood cells in the vaccinated treatment due to less killing and less production, there will be smaller proportion of immature red blood cells. And we could also sequence other known antigens and see if anything else changed. And here are some takeaways from my talk. So evolution, not at vaccine target sites, can have consequences for the efficacy of vaccines. Our results emphasize the importance of parasite defenses against innate immune mechanisms for their role in vaccine-driven evolution. And even if there are no changes detected in the vaccine target sequences, vaccine-driven evolution can still happen. So on that worrying note, I'd be happy to take some questions.